Hi everyone, I am Dr. Deepa American and today I'll discuss on electrophoresis. Today we are discussing in detail on SDS page, native page and immunoelectrophoresis. The word electro, it refers to energy of electricity and phoresis means to carry across. The term electrophoresis means any experimental technique that's based on the movement of charged particles like ions, molecules, macromolecules, etc. in the electric field in liquid medium. It's a physical method of analysis which involves separation of compounds under the influence of electric field. Usually, positively charged molecules, it moves towards the cathode, while negatively charged anions, it moves towards the anode. And the speed of particles that will be directly proportional to the applied voltage, particle charge, but it is inversely proportional to the particle size. Usual molecules or biomolecules used in electrophoretic separation are proteins and nucleic acids. Why we are selecting these proteins and nucleic acids as the common molecules used in electrophoresis? Because we know if we compare like lipids, they don't have much charge. But we know that nucleic acids and proteins have charge. The phosphate groups present in the structure of RNA and DNA that gives the negative charge. So it moves towards the positively charged electrode, that is the anode. In case of proteins, that bears both positive and negative charge. But the pH of the medium or the buffer, it determines the resulting net charge of the protein. We know what is isoelectric pH and what is the condition where the pH is above and below the isoelectric pH. It will act as charged molecules. Therefore, in mildly alkaline electrophoretic buffer, the proteins act as polyanions and hence they move towards the cathode. Moves from cathode towards the anode. And what are the main types of electrophoresis? One is zone electrophoresis and second one is moving boundary electrophoresis. Zone electrophoresis, from the word itself we know that is confined to a particular zone. So in this method an inert polymeric supporting media it is used between the electrodes to separate and analyze the sample. The supporting media used in zone electrophoresis can be absorbent paper, gel of starch, agar and polyacrylamide. The common example of these zone electrolytes are paper electrophoresis, gel electrophoresis like agarose gel that we usually use for DNA and RNA. Then polyacrylamide gel electrophoresis, which we usually use for proteins. Then thin layer electrophoresis, cellulose acetate electrophoresis. And what are the main advantages? The main advantage of the presence of supporting media that minimize the mixing of the sample and immobilization of the molecule after electrophoresis. It makes the analysis and purification of the molecule much easier than the moving boundary electrophoresis. In moving boundary electrophoresis, the electrophoresis is carried out in solution without a supporting media. Here the sample is dissolved in the buffer and the molecules move to the respective counter-charged electrodes. Examples are capillary electrophoresis, isoelectric focusing and immunoelectrophoresis. And what are the disadvantages? The resolution of this technique is very low due to the mixing of sample. This is the main disadvantage. And this is not good to separate and analyze complex biological sample. Instead, it can be used to study the behavior of molecule in an electric field. Coming to SDS page or polyacrylamide gel electrophoresis. SDS page is used for the separation of proteins based on the molecular weight. Usually we know the separation in an electrical field or in electrophoresis that is mainly based on both mass and charge. But in SDS page, we usually use an SDS, and that means sodium dodecyl sulfate, that is a negatively charged detergent, and this negatively charged detergent imparts a negative charge to the whole protein molecule. So there is no difference in the charge and only difference will be in mass. So the net separation that will be made based only on the mass, that is separation is based only on size. In this technique we usually use three type of the buffers. One is running buffer or tank buffer, then stacking buffer, then resolving buffer or separating buffer. Since we are using these three type of buffers, that is called discontinuous buffer system. And let us get familiarized with the buffers and reagents used in electrophoresis. And each reagent, it is having its own role in making this polyacrylamide gel electrophoresis. And first one is NN-N-N- tetramethylene diamine, commonly called as TEMED. It catalyzes the acrylamide polymerization. Here we know that acrylamide, that is a neurotoxic agent that is commonly used in the polymerization reaction, and this acrylamide gets polymerized into its monomeric unit of acrylamide. It get polymerized in this type of reaction. So the catalyst which acts is this TEMED. And for a reaction to polymerization, we should act some external power or energy, something like that. In that, usually ammonium persulfate, that is APS, it acts as a free radical providing agent. Once there is a free radical, then our molecule will be free to attack the next molecule. So this APS provides a free radical, and that way it will initiate the acrylamide polymerization. Now we are having separating buffer, that is stress HCl buffer of pH 8.8, stacking buffer, that is stress HCl buffer of pH 6.8. And these two buffers are very important and their pH are also very important. That we will discuss later. Then we are having glycine, the amino acid, which is a component of our tank buffer. The buffer is tris-glycine buffer with pH 6.3, and here also pH is very crucial. And next one is bromophenol blue, that is a tracking dye to monitor the progress of gel electrophoresis, and this dye contains bromophenol blue. Then beta mercaptoethanol, that is a denaturing agent, SDS, our negatively charged detergent, and also the denaturing agent. Then glycerol to provide that viscosity for loading the protein. Then tris HCl buffer. And for staining or for visualizing the gel, we usually use Commercy Brilliant Blue staining, and in that we use Commercy Brilliant Blue R250, or we can use other type of staining like silver staining also. And silver staining is more sensitive than Commercy Brilliant Blue staining, but easier method is Commercy Brilliant Blue staining. And sodium sulfate we mentioned before, it's a detergent and it is used to denature and provide negative charge to the protein. Acrylamide, it's a monomeric unit used for the preparation of the gel. It will form these type of linear strands, and bisacrylamide, that forms the crosslink for these linear strands. Now we can see a mesh-like structure. So in this mesh-like structure, protein will be trapped and then the protein will be moved only based on the molecular weight. And to analyze the molecular weight of protein, we usually have ready-made molecular weight marker. So why we are using discontinuous gel system? We told we are using three type of buffer system. Why we are using this three type of buffer system, and how our protein gets stacked? Because when we are adding a protein, if we are adding the protein in 1 centimeter length, then the top protein and bottom protein, it should move in the same way. So we have to stack the protein. For stacking the protein, we usually use the stacking gel buffer, that is tris HCl buffer of pH 6.8. And the SDS, and whereas resolving gel buffer, that is the separating buffer, that contains tris HCl buffer of pH 8.8. Both contains SDS. Here the difference is that the difference in the pH, and we know that this tris HCl buffer, it contains chloride ions, and in the case of tank buffer, that contains tris glycine. So they, instead of chloride ions, there it contains the glycine ions. and the pH of this tank buffer is 6.3. And glycine is an amino acid. So most of the amino acid, it is having the isoelectric pH near to 6. So in the 6 pH 6.3, our glycine, which is an amino acid that exists in the isoelectric form, it has very low mobility. So the protein will be stacked between the slowly moving glycine and chloride ion, which can move faster than the protein ion. So it will be stacked in between the chloride and glycine ions. Once the protein sample enters the separating gel, the pH changes from 6.8 to 8.8. At the 8.8, glycine doesn't act as ions, or its isoelectric pH, it had changed. It is not in that range. So glycine, it can attain the ionic behavior and then it starts moving fast. So the protein can also move fast along with this glycine and chloride ions. So now in that 8.8 range, there is no stacking, the separating occurs, and the separation is just based on the molecular weight. And the electrophoresis here, what we'll carry out is vertical gel electrophoresis. It has a two buffer chamber, an upper chamber and a lower chamber. Here we can see the buffer system. This is the instrument. Here what is, this is the gel what we used to cast. This is the tank and the electrode chamber, and this is the power supply unit. Here this is exactly how it looks like while running the gel. This is our tank, and this is having an upper chamber like this and a lower chamber like this, and these both chambers are having electrodes. I will show it here. This is one, it's in the backward position. So this is one electrode, this black colored one, and this red colored one is the another electrode. And here we will pour our tank buffer, that is tris glycine buffer, in this lower region, sorry, in this upper region, and in this lower region. And once this buffer it is being poured, then we can place the casted gel to this system, and then we can clamp this to the gel system, and this upper and lower tanks, it is now connected each other by means of this gel, which is seen in the, which is having the SDS. So this is our connection, and now we can see how we are preparing the gel. Our gel, first it is prepared by this two type of rectangular glass plates are there. One is one rectangular glass plate and the another one is notched one. And in between these two glass plates, we usually used to keep a spacer. You can see this green colored one, that is spacer. So this spacer is in between two type of glass plates. And the lower portion of this one, we will seal it either with agar or some other sealing agent. Then after sealing that, we will start pouring our separating buffer to this lower region. Separating buffer, it contains tris HCl, then SDS, and it's in the pH 8.8. After solidifying the tris HCl, I mean this separating buffer, we can just slightly tilt and see whether it is solidified or not. After solidifying this separating buffer, we usually add the stacking buffer on the top of separating buffer. And before solidifying, we usually add the comb, that is this one. And this comb, it is being placed so that we can, after solidification, we will remove this comb. We can see wells like this where we usually load our proteins. And for loading protein, we usually mix our protein sample with the bromophenol blue solution, and we know that it's contained beta-mercaptoethanol and SDS, two type of denaturing agents. And also additional to that, we will boil the sample so that it will denature completely, and then we can load the sample to the wells. Once the sample is loaded, we will connect the power. We will connect it to the power supply, and our loading die, that is the bromophenol blue, it tracks where the protein it is being moved. When it reaches almost this more than 3/4 position, we will switch off the power supply, and now we can stain our gel either with the Kumasi Brilliant Blue or any other type of staining. So usually we will use Kumasi Brilliant Blue staining that contains this CBBR250 and then glacial acetic acid and methanol. So our gel will look dark blue color, and after that we can destain our gel using destaining solution which doesn't contain the CBB. It contains only acetic acid and methanol. So this is the separation of egg white. This gel shows the separation of egg white. This is partially destained and this is completely destained. Egg white that is separated into different proteins. We can see different bands, and this thicker band that is for the egg albumin. And here this is another purified protein. We can see a band in this region. This is another purified protein. We can see a band here. This is the Sigma staining. And usually we used to add molecular weight marker. We can see molecular weight markers here. That is for the identification of molecular weight. And this will be provided by different companies, and the molecular weight marker of these things are also known. For this one, it is usually this is 6.5 kilodalton, and this is 16 kilo. Sorry, um, this is 14 kilodalton, and this is 20, 29, 66, and 92 kilodalton. So now we can see what is the molecular weight of our protein what we had purified. This is the protein what we had purified. It is seen as band. So this is 6.5, then 14 and 20. So it is near to 20 range. This way we can calculate or we can identify what is the molecular weight of our protein. And also in this gel, we can analyze whether our protein is pure or not. Here I told you this is a crude extract. So we can see many bands, that means the protein is not pure. Here we can see partially some bands, but here we can see only one band, here also only one band, that means it's completely purified. For checking the purity also in this way we can use the SDS page. Also we can identify the presence of disulfide bonds by means of this SDS page. This is if the protein, if we are having a protein and that protein is having 8 kilo and 16 kilodalton size, and after this SDS page, if it is moving as 8 kilodalton, that means somewhere some disulfide has broken and then it was moving into its monomer form. In that way, by comparing the molecular weight, we can analyze whether it is having any disulfide bond or not. So let's see the difference. What is native page and what is SDS page? SDS page, we know that is the denaturing page. Native page, as the name indicates, it is in the native, the protein is in the native stage. That means protein is in some kind of virgin state. It is not in a denatured condition. SDS page contains the anionic detergent SDS and beta mercaptan ethanol and also heating. These three properties, it denatures our protein. But in the case of SDS page, we are not heating our protein, we are not adding SDS, we are not adding beta mercaptan ethanol. So there is no denaturation, and the 3D structure of protein, it's not destroyed and it is well maintained. And the protein will be, since there is no SDS, the movement of protein will be based on both charge and mass. Since it's not destroyed, it's in the native form, so we can analyze the biological activity of protein based on the native page. So, for example, if you want to analyze any enzyme activity of any protein, we can do the native page and then give suitable substrate to that enzyme and see the enzyme catalyzed reaction in the gel itself in the case of native page. Native page also helps to identify the isoforms of protein. Isoforms of protein, they are having same function, but there is very less difference in the amino acid. So very less difference in the amino acid means there is difference in the charge. So that difference in the charge that will be highlighted in native page. So the movement will be based on both mass to charge ratio. So in that case, we can see different bands if it is having isoforms of protein. But in the STS base, that will be shown only as one single band. And now let's move to immunoelectrophoresis. Electrophoresis, now we describe now what is electrophoresis and what is this immuno. Immuno means that is the antigen antibody reaction. So it is a precipitation reaction based on the interaction of antigens and antibodies. It is based on soluble reactants. When here we can see this is the antigen, that is a soluble reactant, and antibody, that is also a soluble reactant. When they come and interact each other, they form an insoluble product, that is a precipitant, and that is being analyzed here. This is the precipitin band after this antigen antibody reaction. It is mainly used to determine the blood levels of immunoglobulins like IgM, IgG, and IgA. And how it is being done? It is usually done using agarose. Usually, agarose we used to do for nucleic acids, but in this case also we are using agarose. The gel, like you are familiar with the glass lights. Like those kinds of glass lights are used for this immunoelectrophoresis. The gel it is being prepared. It contains the well, and to that well we will add the antigen mixture, and then we will allow for the electrophoresis. Then this antigen mixture will be separated based on its charge and mass. After separation, we will cut a well in this area and add antisera to that. And this antisera is allowed to diffuse towards the antigen. And if there is any antigen antibody interaction, we can see the precipitin line. This is one precipitin line. Here the concentration of antigen is less, so the precipitin line is also less. Here the concentration is more, so the precipitin line is also more. You can see here like this precipitin lines. This is also called a rocket immunoelectrophoresis because if the concentration is more, we can see like a shooting up of the rocket, like precipitating band. So it's called as rocket immunoelectrophoresis. And what are the applications? It is usually used in clinical laboratories for detection or presence or absence of proteins in the serum. It is also useful in determining whether the patient produces abnormally low amount of one or more isotopes of IgG, which is usually characteristic of immunodeficiency diseases. It also can show whether the patient overproduces some proteins like albumin, immunoglobulin, or transferrin, which are indicators of some other diseases. The main advantage of immunoelectrophoresis is that antigens can be the amount of antigen, or it can be quantified in using the identified serum. If they By seeing the precipitating lines, we can quantify how much antigen is present. So hope you have gone well through the session. And if you have any doubts or queries, please contact. Thank you.